Okay, so now we're going to talk about antibiotics, just in general. You'll also cover this in different pharmacology lectures as well. So the first thing to recognize with respect to antibacterial antibiotics in general is whether they are broad spectrum or narrow spectrum. And so our broad spectrum antibiotics are things like penicillins and other beta-lactam antibiotics. And this is because they're going to be effective at targeting the peptidoglycan by targeting the penicillin binding proteins. And keep in mind that penicillin binding proteins are bacteria origin, and they are necessary for building the peptidoglycan. So in essence, these broad spectrums are knocking out the enzymes that are required to make the peptidoglycan. So they are going to be broad spectrum. Narrow spectrum are going to have a limited effect on different bacterium. And so vancomycin is considered a narrow spectrum. It can affect a lot of gram-positive bacterium. It has a more difficult time with gram-negative bacterium. And so vancomycin is going to target the D-alanine cross-linking. Okay, and some bacteria don't even use D-alanine cross-linking, and so they would be more resistant to vancomycin. Vancomycin is typically going to be almost the last resort for severe staph infections that are resistant to some of the more broad spectrum antibiotics. Now, another narrow spectrum is fidaxomicin, and this is going to be effective at treating Clostridium difficile associated diarrhea. So it treats C. diff. And so this is, again, another narrow spectrum, but it's effective at a very important hospital-acquired infection. Now, the other thing to keep in mind about bacteria or antibiotics is that they are going to either kill the bacteria and be bactericidal, which is our penicillin, cephalosporins, vancomycin. They're targeting a component of the bacteria that the bacteria cannot live without. And then you have the bacteriostatic, and these are going to inhibit the growth of the bacteria. So these are going to do something with metabolism. And so they're not going to basically kill the bacteria immediately, but they're going to slow it down. They're going to inhibit the growth. And keep in mind that antibiotics are also going to have the added benefit of working alongside our immune system. So as the bacteriostatics are inhibiting the growth, our immune system is going to be trying to eliminate the bacterium as well. So there are several reasons for combining antibiotics. Of course, to prevent resistance, the idea that a bacteria colonizing a person is going to have multiple resistance is not highly likely. And so by giving combination of antibiotics, you're able to eliminate these resistance to penicillin, for example, as you give vancomycin. You can also have synergy with killing, and so a severe infection might require more than one type of antibiotic to get it under control. So synergism is combinations that enhance the bactericidal activity of the combination rather than just the individual antibiotics alone. And of course, you want to avoid antagonism where one antibiotic is going to interfere with the activity of another. And so this is just a picture to show the different types of antibiotics that are currently available. We have the bactericidal, and these are going to kill the bacteria. They target the cell wall synthesis, and so of course a bacteria that cannot survive without its cell wall. And then you have the bacteriostatics. So you have your antimetabolites that are going to target sulfur. You're going to have targeting of your protein synthesis. The protein synthesis, of course, will be bactericidal over time because your bacteria need to make their own proteins. DNA replication is going to be targeted by your quinolones. You have your RNA synthesis by rifampin. Chloramphenicol is going to, again, target protein synthesis. It targets a different part of the ribosome than your tetracyclines. But anyways, these are all going to be effective at not only slowing down bacteria, but eventually helping getting the bacteria under control. Now, our big class of antibiotics are our betalactamase. Betalactams. Betalactamase is a bacteria enzyme. That targets the beta-lactam antibiotics. And these are, of course, your penicillins, plus the like. And so what you have to keep in mind about the beta-lactamases is they're going to be very specific. There's going to be a penicillinase, that's a beta-lactamase that inhibits penicillins. There's going to be a cephalosporinase. There's going to be a carbaminase. And these are all going to specifically target specific classes of antibiotics. They are generally referred to as beta-lactamase enzymes that are in the bacteria. And so where do these bacteria get these beta-lactamases? They're going to get them from those processes we talked about through plasmids, acquiring plasmids, by your bacteriophages, bringing in genes. Things like that will transfer all of those genes to the bacteria. So antibiotic resistance. So there are, of course, going to be the exchange of genes. And so this can lead to acquiring resistant genes, such as the beta-lactamase. And then there is going to be gram-negatives. They tend to be a little bit more antibiotic resistance because they can alter their pores in their outer cell wall. And they can prevent antibiotics entering the inner membrane space. So if you'll remember the structure of a gram-negative bacteria, the peptidoglycan layer is in between the two membranes. And so in order for our beta-lactam or our peptidoglycan targeting antibiotics to get access to the peptidoglycan, it has to be able to transverse that outer cell wall. And so gram-negative bacteria can stop many antibiotics at this point. Your gram-negative bacteria can also pump out, which is egress, antibiotics. And so this is just another function of your gram-negative bacteria. Now, all bacteria can change their enzymes. So they can alter those penicillin binding proteins. And this would be through like point mutations. And of course, this would be random, but it would be selective. So bacteria within a quorum that creates a penicillin binding protein that no penicillin can no longer bind to will become resistant to penicillin. And so keep in mind that the penicillin binding proteins are encoded by the bacteria. Okay, and so that's our short discussion about antibiotics.